So again, Richard would like to thank the organizing committee for inviting him. Unfortunately, about a month ago, he found out that he couldn't attend, so he asked me to fill in for him. So my name is Francois Belzile. I'm the last line there. And so I've been working with Richard on this type of work for a few years, and I'll be trying to tell you some of our work on silicon transporters and some of the benefits that this brings to plants. So we've heard a lot in the last hour or so about all the different beneficial effects of silicon, both in biotic and even in abiotic stress relief. So I won't belabor the point. I think we've heard and seen a number of these pictures showing some of the prophylactic effects of silicon in helping to protect them from attack from many different pathogens. We've seen both some effects on foliar pathogens, and this is a case where this is a root pathogen of cucumber, again showing quite clearly that the silicon amendment was beneficial in protecting this plant. Now, as was just mentioned earlier, we can differentiate plants into different groups based on whether or not they tend to accumulate a lot, a bit, or intermediate levels of silicon. And so obviously this is a fairly crude division of plants into these three different categories. And so basically this helps explain sometimes some of the less spectacular results that have sometimes been observed with plants that are not known to accumulate a lot of silicon. So if they don't accumulate it, you would find it hard to see that many benefits. But obviously, in the high accumulating plants, we've heard a lot about rice, and we'll be talking somewhat about the horsetails today, and you'll see that there are some pretty interesting things going on. So until maybe 5-6 years ago, until Dr. Ma's paper came out, nobody really knew how a plant could take up silicon from the soil solution. And I guess when his team came out with this silicon transporter in rice, it helped illuminate a number of phenomena. And so basically the model that this Japanese group has been elaborating over the last number of years basically helps to explain how silicon in the form of silicic acid can be taken up from the soil solution and distributed up into the plant. Here's what looks like silicic acid. And in rice, they've described two of these transporters, which are membrane proteins, basically, that will allow the silicon to traverse the membrane. And so basically, in the rice system, both of these seem to be required. One here is what we call an influx transporter, which helps take the silicon from outside the cell in the cell. And then there's this other one, LSI2, which would take it from inside the cell and pumping it out. And so you get this series of transports that allows the silicon basically to make it through all these different cells and up into the xylem and then gets transported up to the aerial portion. And so that's been the model that a lot of people have been working on. And when you do some phylogenetic analysis and looking at the different genes that look like LSI1 in other realms of the living, you find that they all belong to this NIP3 family. So this is one big family of genes that are in code for proteins involved in transport. And so these are where all the known silicon transporters lie. So they have these specific characteristics. So you'll be hearing a lot from me about these NPA domains or NPA loops. These are short amino acid sequences that are highly conserved in the sequence of these proteins. And then there's this other thing here that's GSGR. This is the amino acids that form a pore. And it's through this pore, basically, that the silicic acid is transported. And all known silicon transporters today have this same pore. And so it seemed to be a very important component of this protein. And thanks to whole genome sequencing projects in the last few years, there's been a few of these ferns and mosses that have also been sequenced. And when people look for homologs of these genes, there are a few things that look similar. But you'll notice here that despite the fact they have these two NPA loops, they have quite a different set of 4 amino acids that would potentially form a pore. So something quite different. And so we've been interested, obviously, in working on a wheat system. You'll understand that in Canada, rice is not a very big crop. And so wheat was a more interesting model system to study this. And so, again, here another example where the powdery mildews are kept in check thanks to the silicon. And so what was interest to us was obviously trying to find some wheat transporters that would be doing similar stuff to the rice genes. And so basically building on the sequence homology that exists between these two closely related grasses, we were able to design some primers to amplify up portions of genes that looked like LSI1 in this case, and through subsequent analysis to describe the entire gene. And when you line up the DNA sequences, you're not very surprised to notice all these red segments which are identical, meaning that these are very similar genes in terms of their DNA sequence. And if you look at the protein sequences, they're also extremely similar. So on a homology basis, we'd like to say that 81% of these amino acids are conserved. And you find the same functional elements such as these NPA loops or motifs that are these 3 amino acids. And the highlighted in red, these are these 4 amino acids that in the 3D structure of the protein end up forming the pore, or the key, determine the specificity with which different solutes can go through this pore in the cell. And so one way to analyze these proteins for function is to actually use a heterologous system. Here it's the frog eggs, and so basically what you can do is you can inject these eggs with RNA coding for the gene that you're interested in. The protein gets synthesized inside the egg, and if everything goes well, the membrane, the protein lodges itself in the membrane and will confer the ability on these frog eggs to take up silicon in this case. And so it's a very nice system to test whether or not a candidate gene does indeed code for a protein that allows silicon to be taken up into these frog eggs. And so this is a slide that demonstrates basically such injections. And so at time 0, so when nothing has happened yet, you see that the control and the rice and the wheat, there's not much silicon going into the cells. But after 30 or 60 minutes, you see that there's a strong increase in the silicon that's been taken up by these frog eggs, thanks again to the fact that we've allowed these eggs now to have these transporters that allow silicon to enter. And interestingly, you can see that both the wheat and the rice had a comparable capacity to accumulate silicon in these frog eggs. So where do these wheat transporters fit in? Well, you won't be very surprised to see that they fall within the same broad category that was already well known, and again, very close relative to the barley, this Hordeum vulgare, and so the close relative of wheat. Again, no big surprise there. So it was nice to see them fit in the family there, but again, we wanted to go a bit further, and when we thought about, well, what should we study? We thought, well, Horsetail would be a good model. We've heard earlier that it's king of silicon absorption. And in fact, in our case, when we grow these in solution in the greenhouse, if we don't add silicon, the thing doesn't grow. And so this question of essentiality, well, if the plant doesn't grow, I think it's essential for the plant. So in this case, that certainly is the case. And if you do some microscopy to sort of see where the distribution of the silicon goes in these plants, in wheat, they have these nice rows and files of cells where silicon tends to accumulate. So the more the intense the color, the red here is where it's most intense, so the richest in silicon. And if you compare that with the horsetail, it's pretty much a monstrosity of silicon accumulation, and so it certainly helps to explain why this plant can accumulate such a very, very high content of silicon inside as part of its dry matter. So, horsetail. It's a monster in many ways, so has a huge genome. Genome about the size of wheat, 5 times human genome, just to give a reference. Call it useless. Nobody really wants to grow horsetail. It's a weed in many cases. And so, unsurprisingly, well, there's not much information in sequence data banks when you look for horsetail sequences. We were basically starting from scratch, and so we thought, well, why not use the same approach as we used in wheat and see if we can't fish something out using homology-based methods. And so we tried with these PCR approaches, and that turned out to be, after many months of fruitless labor, to yield nothing. And so I thought, well, let's have a look by southern analysis, and maybe we'll be better able to see some weak homologies, and turned out that that was not the case. So this is a wheat cDNA probe that's being applied onto genomic DNA. You can see that the rice here, homologs, the barley are detected without any problem under these conditions, and horsetail is just a blank. And so that tells us that if there are silicon transporters in horsetail, they must be quite different from the ones that we're finding in cereals. And so we decided to use a different approach. I'll let people guess who's who on this. So we had discussions about the usefulness of going to next generation sequencing to do some analysis of what was going on in horsetail. And so basically this is where we extracted RNA from roots. Again, this plant does not grow if we don't provide it with silicon. And by analyzing the RNA populations inside the roots, I'll spare you some of the technical details. The important part is down here. We assemble what we call contigs. So in the context of an RNA analysis, what we call a contig is basically corresponds to a gene, a transcript, a cDNA, and mRNA. So we're finding basically in the root of this horsetail plant about 7,000 different genes being expressed. And we then use conserved protein domains from other known silicon transporters and try to probe this collection of RNAs to find candidates for these transporters. And among a certain list of things that looked like they could be transporters, we had this one candidate that was on top of our list because on a number of different characteristics, it seemed like a very plausible candidate for this role. And so these are NIP-like aquaporins, so they belong to this family of aquaporins that are proteins that facilitate the import of solutes. And it's, again, this specific family. And so we were interested in probing how this family fit into the broader picture. And so in this case, what you'll notice is that all these horsetail putative transporters, I guess, at this stage, fall into this group here, which is what we call the NIP2 family, whereas all the other known are up here in this NIP3. And so we have, from a phylogenetic point of view, a group of genes that fall in a separate category. So obviously that's going to be somewhat novel. And you'll notice here these famous NPA loops and the GSDR pore. Down here we still have the NPA loops in many of these, but we have a new set of 4 different residues that we call the star pore. We like to cite them in that order, and it's the order in which they appear in the sequence, so we didn't make this up. So we have this group of very closely knit homologs that have the looks like they could be transporters, but we really need to test functionality. And so we did some structure analysis, so using some softwares that can try to describe what the protein looks like when it's properly folded. We get these models that show that there are these transmembrane domains, and these are required for this thing to be present in the membrane and to play its role as a transport protein. And these are the 4 star, these 4 residues. And this software does basically a prediction, says among the solutes that it knows what likely solutes that would be most compatible with this pore. And it turns out that actually salicylic acid was #1 on the list. And so it was very satisfying to see that it came out as a good theoretical candidate, at least. And so obviously this is all modeling. It's putative, so we wanted to test this in a living cell. And so we went back to the med lab to do some work on some more frog eggs. And so here's the results that we got with these horsetail transporters. So in this case, we tested 3 of these, so NIP3.1, 3.3, and 3.4. And what you can tell is that basically they do indeed allow for quite a significant amount of silicon uptake. And so all 3 of these certainly were effective in allowing the import or the uptake of silicon, but 2 of these, so 3.1 and 3.4, seem to be particularly competent at achieving this and basically seem to lead to a greater silicon accumulation in these frog eggs than even the rice transporter that we've spoken about abundantly. So then frog eggs, they're nice, but they're not really a plant, and so we decided to put this into a plant. In this case, chose Arabidopsis to do this heterologous work. And so we took basically the NIP3.1 cDNA from horsetail and attached that to a constitutive promoter that's going to be expressed in most cell types in Arabidopsis. And so when you look at either roots or leaves, well, you'll see here presence of the mRNA, so the gene is being expressed in both roots and leaves under this promoter. And what happens then to silicon uptake? Well, this is basically the wild type Columbia Arabidopsis that is not transgenic. It does not have this horsetail transporter. And you see that when you do add silicon, it is capable to take up a bit of silicon. So as one of these dicots, that's a low accumulator. It doesn't do spectacularly well. And with the horsetail transporter, you see that there's a much higher level of accumulation of silicon that's made possible thanks to the introduction of this silicon transporter. And so when we go back to thinking about, well, why is it that we couldn't pick this out earlier when we were doing these attempts at cloning through homology-based strategies? Well, you can see the answer here. Look at the degree of homology between the grass, so rice and wheat, and the horsetail. It's below 40%. So this is, again, it's a protein that's got a very similar structure in terms of how it forms transmembrane domains and how it can create a pore, a hole for the silicic acid to penetrate. But evolutionarily, it's very, very, very far removed or different from the rice and wheat. And again, this probably reflects the fact that this is a very, very distant relative of, I guess, monocots in this case. So where does it fit in the overall picture that we've been developing? It turns out to be on quite a different branch. So I mentioned that instead of fitting nicely in the NIP3 group where all these other ones are, it fits on quite a separate branch up here in these other NIP2s. And those that have been characterized in this group are not silicon transporters. So there's something doubly unique or novel in residing in this branch and still being a silicon transporter. And it has these novel characteristics at the level of its pore, which is, again, the first in its kind have been described in the plant kingdom. And so we'll leave to others to decide whether or not it should found a new family and call it NIP5. This phylogenetic analysis is not exactly our area, so we'll leave more competent people decide whether or not it should form a new branch and have its own label in this tree. But we'll leave it at that for now. And so basically, for those of you that would like to get a more complete rendition of this story on the cloning of these horsetail transporters, the paper came out this month, so you'll be able to look it up in the Plant Journal. And so what this has brought us basically is the discovery or the description of a novel group of silicon transporters that have this ability to import silicon, which seems to be even higher than the one that was previously described in all of the angiosperms that have been looked at before. And it may contribute, so this exceptional capacity by these transporters may help explain why horsetail is such an exceptional absorber of silicon. And it's obviously a very nice and powerful tool to help us try to understand how silicon is absorbed in plants and can do all the beneficial things that you've been shown that it can do. So with that, I'll stop there, and I hope we have, yes, we still have a few minutes of coffee break.
Yes, we do. And this is supposed to be